And now we're to 29. This is the last chapter in the book.
That's a lot of chapters.
Chapters
And a
just to
lot of-
I know, right? Just
It's
to
what?
cut it out.
That's a lot
Like
of
a congratulations
chapters.
chapter or something.
Yeah, that's a lot of reading. It's a pretty thick book altogether. And I can tell you that when I had been away from anatomy for almost 40 years doing surgery. And I came back and started teaching here at PCC, and I made a commitment that I would read the entire book, every page.
Back up.
That took a lot of time. I was doing a lot of treadmill time and stationary bicycle time and trying to stay somewhat in shape. And I found that I could read about 2 pages an hour. And that wasn't because I was distracted. I mean, it just takes a long time to read this stuff. And that's the way scientific reading is. But anyway, here we are, we're almost to the end, and we'll talk about the last chapter now. I gave you, as far as the chapter is concerned, we won't have a quiz over this chapter. There are some questions on the final written exam. And there are some from the lab, in the lab practical, about meiosis, mitosis, and this sort of stuff. And Samantha, here you go. And then we will have a quiz on Wednesday on the reproductive chapter, which is really a long chapter. I always think of it in 2 parts, male and female, because it splits that way. And then when you come in for your final exam, which is next, a week from today, remember to bring your lab sheets, the tear-out sheets that we've done for the first 2, bring those in, and bring in your pedigree problems, and bring in your name tags, your little tints. So that's a lot of points on the last day, besides the 200-point practical, or written exam, and the 75-point practical exam. But you got to bring all that stuff. So, maybe we need to make a list on the board in the lab. Yeah.
Blood draw donation.
And the blood draw donation, I'll pass a thing around, maybe today in lab, and ask each of you to just sign it if you made an attempt or if you donated blood. It's on your honor. Way, here's your, it's on your honor, and I don't really care whether you were able to give or not. I just wanted you to try, experience the whole thing. Yes?
Where is our final exam? Is it in
Same place.
Same
yeah,
in here?
Right here in
Okay.
this room, same time, 1 o'clock. If you want to come a little early, there's several of you, especially those of you that English is your second language, you're more than welcome to come in maybe 20 minutes early to this room, and you can start. Personally, I don't think it takes an hour and 20 minutes, but if I give you, an hour, and if I gave you 2 hours, there would still be some of you sitting here at the end. If I gave you 6 hours, there would still be some of you sitting here at the end. That's just the way you are.
Just to be sure. Read it over.
Yeah, read it over, change your right answers into wrong answers a few more times. That's just the way you are. That's why I just think we ought to just do it an hour and 20 and call it good. But I'm trying to be nice to some of you who angst so much about these tests. Okay? All right. Development and inheritance. These guys both have a set of DNA. It's probably 99% the same. Probably 99.5% the DNA in a cat and a mouse is exactly the same. This one has, he gets bigger. They both have hair. They both have 2 eyes. They both have 2 ears. They both have 4 legs. They both have tails. Think about it. I mean, they both have a digestive system, liver, gallbladder. Their DNA is pretty darn the same, pretty much the same, and that's the same with you and your kids, and you and your brothers and sisters, and so forth. And if I had one piece of advice for you today, it would be don't ever go to a rave in Oakland, California. Today they have put out an appeal for all the parents and all the relatives of all the people that they think are burned up in the building to please save their DNA. That means save their brushes, their combs, their toothbrushes, trying to be able to match DNA with bodies, which is, there was a fire that's killed 38 as last I heard, and they're thinking it may go to 50.
It
They
was a
don't,
rave?
yeah, it was a rave gone bad, I guess. My mother made a comment to me, and I think about it every time I hear about something like that happening. And if my mother told me this one time, she told it to me 100 times. She said, Hal, nothing good happens after midnight.
I say that all the time to my
Huh?
kids. I say that all the time to my kids.
Yeah, nothing good happens after midnight, and by God, it's the truth. There's not anything that you can't get done before midnight that you can get done after midnight.
Is that
But
why-
by God, it seems like, and I mean, every morning I listen to the news as I drive into school or wherever I am, I listen to the news, and all the shootings and all the robberies and all the bad crap that happens in our society happens after midnight.
Is that why it's Boeing 1201?
I don't know. Maybe
Nothing good
that
happens after midnight, ladies and gentlemen.
So, let's talk about this development business a little bit. First of all, when you started, we were all one cell, and that one cell is called a fertilized egg or a zygote. And remember, it's just one cell. And that one cell then starts cleaving and starts dividing, and one cell goes to 2, goes to 4, goes to 8, goes to 16, 32, 64, and so forth. At some point, however, those cells have to start differentiating. And some cells have to turn into muscle, and some have to turn into nerve, and some have to turn into our skin and our brain and all these organs, these 28 previous chapters that we've talked about. And as the movie talked about the other day, it's because of selective changes in the genetic activity. Certain proteins are turned on, certain genes are turned on, certain genes are turned off, things develop and things digress. We've talked about fertilization. It's also called conception. It's when development begins. I have a really hard time with the political arguments that go on about when life begins because it's very complicated. And one thing we know is that over 50% of fertilized eggs are lost. And it would be sad to think that half of all the conceptions, half of all the lives automatically got to go to hell, I guess. I guess that's what you believe. If they didn't get a chance to get right with religion, hell's filling up fast at that rate. But anyway, I struggle with that stuff, and I think it's important that you all kind of try to think about all of this stuff in the context of what reality really is. And it becomes hard. But fertilization is really when development begins, even though the cells don't start to differentiate right away. Embryological development occurs in the first 2 months after fertilization. We call that embryology. When I was in medical school, we took gross anatomy, which was one of our classes. We took histology, which was one of our classes, and we took embryology, which was one of our classes. And in that embryology class, we studied this first 2 months of development, cell by cell, under the microscope and through lectures and so forth. They call it fetal development, starts at about the 9th week. In other words, it doesn't turn into a fetus until the 9th week. These are all just divisions made artificially by humans, I think, so they could be splitters instead of lumpers. And so we call it, beginning at the 9th week, we call it the fetal development. Prenatal development involves before birth, so it involves both the embryological first 2 months and then the fetal development stages. Postnatal development is what happens the instant you're born. I guess natal is birth, right? Nater, birth. Starts at birth, continues on to maturity. I'm still going through postnatal development myself, and all of you are too. It's called aging as we get older, I guess. A couple of words you need to understand. Inheritance means the transfer of genetically determined characteristics. In other words, you do give your craziness to your kids. And we all pass on things to our kids, our hair color, our size, all that kind of stuff. Genetics is a study of the mechanisms responsible for that. You've all probably had some Mendelian genetics. Mendel was this, what was Mendel?
He was a monk.
He was a monk. And where did he, what did he study? Peas.
Sweet pea.
And I don't know if any of you ever thought about this, but he was like the luckiest guy in the whole world. Because what he did was he picked traits that were truly recessive and non-recessive. You know, he picked wrinkled peas and tall plants and short plants and long seeds and short seeds and
Pink flowers and white flowers.
pink flowers and white flowers, all those things you remember hearing about, and one of them was always dominant and one of them was always recessive, but we also know that almost all traits are not that way. They don't really work that way. But he was lucky and he picked those. The other thing he did, which was different than everyone else, because he was not the first guy to sort of figure out what he figured out. You know what was really special about that guy?
He made it popular.
No, he didn't. He was long dead before it was ever discovered. What he did was he took notes and he counted and he wrote the numbers down really carefully. And if you want to read something that's really interesting, go back and read his papers, because he'll talk about how he planted 317 of this kind of seeds and he got 75 or 78 of this, and he got 225 of that, which was 1-to-3 ratio. And his ratios were all amazingly close, but the main thing was he took really good notes. And although other people were proposing these ideas of this mystical genetic material that they had no idea what it was, they had no idea it was chromosomes, they had no idea it was DNA, they had no idea, it was just a magical something that carried traits in those days. But he took very careful notes, and that's why it all got named after him. So whatever you do in research in life, take good notes. It'll allow you to go before the Nobel Committee and get $1 million. The thing was, you can't get money from a Nobel Prize if you're dead.
Well, you know, according to Mythbusters, the difference between screwing around and science, documentation.
Documentation, that's about the truth.
Only difference.
So anyway, but Gregor Mendel was a pretty interesting dude, and I guess it just shows you that he didn't really have very much to do as a monk, but he was able to count his peas very carefully.
Well, that's important.
It was important,
You should know how to count.
yes. Anyway, so fertilization, we'll talk about. Fertilization is the fusion of two haploid gametes, and that means you're bringing a male sperm that has 23 chromosomes to a female egg that has 23 chromosomes, and they get together, and that produces a zygote, which is also called a fertilized egg, and it contains 46 chromosomes. And remember that out of those 46 chromosomes, 44 of them are what we call autosomes, which means they have all these different traits on them, and then 2 of them are the sex chromosomes, and they can either be 2 X's, and if they're X's, they're pretty big chromosomes and they have a whole bunch of genes on them, or they're Y chromosomes, and Y chromosomes don't have much of anything on them except a little bit of DNA that makes you make some testosterone, which was what makes you a male. But everything else is carried on all the rest of the chromosomes, and certainly of all the chromosomes there are, the least important one is the Y.
That explains a lot.
Right?
The spermatozoon, you know, that's the thing that delivers the male chromosomes. It has to travel a large distance. It's pretty small, it's very efficient, it's very highly streamlined, it's made just to do what it does. The gamete is a generic term. Gamete means sex cell. And usually the female sex cell is the one that brings all the organelles to the party. The inclusions, the nourishment, all the cytoplasm kept getting put to that one side in the divisions, and the polar bodies kind of went off, got lost with their little bits of DNA on them. And remember that that gamete, that egg, has to live almost a week on its own, just floating around. You know, it has to get thrown out of the ovary into this cruel, cold abdomen, and it hopefully gets sucked up by this fimbriated end of this fallopian tube, and then it gets floated down this tube for a few days at least, and finally it may meet up with a sperm or not, and then it has to continue floating on down for another 3 or 4 days as it starts its dividing thing, and then it implants into the uterus, assuming it gets down and implants in the right spot in the uterus. So everything it leaves the ovary with, that has to sustain it for a whole week. Now it's going to pick up a little bit of sugar along the way. The sperm, of course, doesn't have anything. He has to pick up sugar all the way, that fructose stuff. And there we go. So fertilization usually occurs within the first day or so. It usually occurs in the uterine tube. The secondary oocyte, which is what was thrown out of the egg, out of the ovary, only travels a few centimeters, but the spermatozoa has to travel all the way from the vagina all the way up to usually within somewhere in the ampulla of the tube is where the ovulation occurs normally. We talked about capacitation when we were talking about the final maturation steps in the sperm. It really has capacitation is really the last stages in preparation of the sperm, and it really doesn't even happen. It doesn't become fully mature and ready to go until it comes in contact with the secretions of the seminal glands, which means it doesn't get really activated until ejaculation occurs, and then it has to fight off the bad conditions in the female reproductive tract, the acidic conditions and so forth. A couple of enzymes you need to know about. One is called hyaluronidase. It's an enzyme that breaks down the bonds between the follicle cells. So it enables, when the hyaluronidase is released by the sperm, it makes the follicular cells get out of the way and split apart, so at least the sperm can get into the surface of the oocyte to get in there. And then it needs this acrosine, which is another enzyme, which is required to break through that last layer. The zona, right?
Corona radiata,
Zona pellucida, I think so. I don't know, ask Daisy, she probably knows, I don't. But anyway, the spermies have this little cap on their head up here, and in that little cap is where these enzymes live, and they are needed to penetrate both of them, is the answer. Corona radiata, zona pellucida, to get down to the surface. And once that happens, then the sperm membrane merges with the membrane of the oocyte, allows the contents of the sperm, which is basically just the chromosomes, to enter. And it's only at this point that actually the oocyte matures all the way to where it needs to be. And this is where it finishes meiosis II and really becomes an ovum. So I've always thought that was weird. I mean, you talk about holding off till the last minute, you know. I mean, this is worse than having to get your hair done before you go out. They're going out and their hair's not even done.
We'll do it in the car.
You know, they'll do it in the car, I guess, because they don't even finish meiosis II until they've already hooked up with the sperm and they've already been fertilized. So that's pretty long.
Waiting on a woman.
That's, you know, that's the way it is. Anyway, so that, once they are both together, then I think is one of the most amazing things that ever happens, is because you've got this ovum, this egg, that had this set of DNA, 23 chromosomes in it, and along came this little spermy thing here, and he got in, and now his DNA is sitting over here somewhere, and he's got 23 chromosomes with it, and they've got to get together, and they've got to not only get together, but they have to then undergo mitosis. They have to split that first time, and they haven't ever even been to the party together before. And now suddenly you've got these two sets of DNA dumped into the same egg, and they have to all line up, you know, and do the metaphase thing, and get ready to line up and split. You remember, that's where this thing called independent assortment comes in, where you get half of your chromosomes from your daddy and half from your mommy, but you may get your daddy one for this trait and mommy one for that trait and so forth. It's all random. And because it's random and because there's 23 of them, and the odds are 1 out of 2, that, you know, like for every one, so it becomes 2 to the power of 23 as to the number of possibilities there are as to how they line up. That's a lot. That's a lot of possibilities. And then when they start doing that huggy thing and exchanging some genes, yeah, it gets even more complicated. We talked about that before.
Crossover and
That crossing over doesn't really happen at this stage, though. I'm getting myself ahead of myself a little bit there. That happens earlier in meiosis when we were when we were creating the egg and the sperm. All right, so these acrosomal caps go through here, and the oocyte finish. I guess I've gotten backwards here. Here's these two pronuclei. Here's the male and the female pronucleus. They both migrate to the center of the cell, and then the two of them kind of merge together. And this is an interesting word, I think, called amphimixis, which is basically the fusion of this female collection of DNA, chromosomes, and this male collection. They come together and get ready to, and now they have 46 chromosomes in there, but then they still have to really merge themselves together and get ready to divide the first time. The first few divisions are pretty simple because they are simply divisions. It's strictly mitosis. There's no meiosis involved here. And like I said earlier, 1 cell goes to 2, goes to 4, and it's not till later that the differentiation starts. A schematic picture, I think, of an egg surrounded by a whole bunch of spermies trying to get in. Here's the ovulation. Here's what's keeping the guys from getting in. There's this zona pellucida around the edge, then there's this corona radiata of all these follicular cells, and that's where it's at this point. Once that sperm gets in and becomes this, the second half of the things here, then the female egg, the egg, finishes her meiosis 2, throws out the second polar body, so that now she's down to 23 chromosomes, and she's down to n number of chromosomes. Mating up with this guy here, he's got another n, and so now they're 2n, and they're ready to start lining up. Here they are lining up, 23 and 23, and then they have their little spindles come in here and line up, and then all the chromosomes condense from being chromatin, right? Remember chromatins, when they're all stretched out, they condense into chromosomes, then they all line up and do the do-si-do, and half of them go one way and half of them go the other way. And then you get these little guys, they call them blastomeres. That's because we just don't have any other name for them other than blasts. You remember blasts? Remember?
immature cell.
Yeah, blast means like osteoblasts and chondroblasts and all those. They're
It's the
they're
early
just
version.
basically immature cells that are like stem cells. They can turn into several different things. And that's certainly the case here as well. So they line up like this and they turn into these guys, which are called blastomeres, which is a pretty unimpressive name. And then they continue to split for a while. And then from the movie the other day, you remember it got to be about 100, and then it sort of got hollow in the middle, and we called it a blastocyst. And we'll go from there. Yeah.
Okay, so I just want to be clear, when the sperm actually does enter the egg, it's still considered one cell, even though two cells converge.
Yeah.
Okay, and that's just because the sperm sort of just releases its genetic material, and then does the rest of it die, or does the egg use?
Well, he doesn't have anything much. You know, he's got a few mitochondria that whip his tail, and he's got a little cap that had some acrosomal enzymes, but those all got released before he got in. Not too much to the guy, other than those chromosomes. Would I say the rest of him goes off and dies? No, it just becomes incorporated in the membrane, becomes incorporated into her membrane, into the cell membrane around the whole thing. First trimester, second trimester, and third trimester. I was going over the exam the other day, and I think the key words in these definitions right here, the key words are that in the first trimester, the rudiments of all major organ systems appear. In other words, it sort of starts looking like an eye. There's a dark spot. It sort of looks like an eye. There is a beating structure which is going to become the heart, but it doesn't really have much form to it in the first trimester. But there's something there, and it's contracting. And I think that's the important thing. The rudiments of all major, like rudimentary organs, are there. They don't necessarily work, and they don't look anything like they're gonna look, but the rudiments are all formed in the first 3 months. And that's why, and you've heard me say before, or at least a lot of you have, is that, you know, if you're ever thinking about getting pregnant, start taking your pills right then. Start taking your vitamins, because by the time you know you're pregnant, you're probably going to be halfway or more through your first trimester, and so most of the baby's structures will have already formed.
Including the neural tube, which you need folic acid for, so you don't get spina bifida.
Right. So you got to, all that stuff that you want to be prepared for, you got to start even before you know you're pregnant. It's not like you wait until you went to the doctor and he says, "Yep, you're pregnant. You're 3 months pregnant now." It's too late, because all the organs are made by then, and if they're not being made properly for lack of some nutrition thing, you're out of luck. You blew it. If you continue to snort cocaine, if you continue to drink booze, and then you find out you're pregnant, you know, you kind of hosed that little baby in there that's no bigger than a poppy seed. But that's when he needed the most care of all, was in the very early stages of pregnancy, and you didn't even know about it, right? Second trimester, and by the end of the second trimester, which is 6 months of your pregnancy, the fetus finally looks like a human. It's not one you'd want to claim, but it sort of does look like a human. Body shape and is very out of proportion. The head is huge, the body is small,
We're calling ours
things like that. Third trimester is nothing but everything getting bigger, rapid growth, really starting to steal from mother, stealing all her fat, all her food, everything the baby can get in order to grow, and soon then it's going to start wanting to get out. Cleavage is a term simply for this big sequence of cell division. This is the 1 to 2 to 4 to 8 things. And then after it gets to about 150 cells, I believe, it gets hollow, and so it becomes like a basketball, which is empty with fluid in the middle and cells all the way around. And that's about the time when the blastocyst is now mated all the way down the fallopian tube, It's laying up against that uterine wall that you remember was growing all those special glands and getting really juicy and really getting excited and waiting for that egg to come along and try to implant in it. And so that all happens at that stage there. Implantation is a critical period of time for the little one. Let's just call it the little one. It's not a fetus yet even. But the blastocyst has to erode into the endometrium. And in order to erode into the endometrium, it really does just grow into it. And by the way, we don't really talk about it in this course, but there are a number of cancers that occur made of the placenta. In other words, the baby keeps eroding and keeps eroding, and it doesn't, it doesn't get the memo that it's time to stop. And the different choriocarcinomas of the chorion and the different membranes of the uterus can go crazy, and women die of cancers that were started by the implantation of the baby into the wall of the uterus. So it's a difficult time, and think about it. If you think about it, it makes sense, because here's this thing that comes along, it lands on this uterine wall, and the uterus has to sort of say, okay, here, just grow into me. And it does, and it just starts growing and growing, and you like to think it's going to stop at the right spot and not grow too much, but sometimes it doesn't. So it gets very complicated here with this implantation. And as it implants, then the idea is not just to implant and live eating into the muscle of the uterine wall. It's really made to cause a placenta to form. And the placenta is what's going to supply that fetus for the next, 8.5 months. And placentation is a fancy word for saying a lot of blood vessels are going to pour in there, and a lot of blood vessels are going to form the maternal half of the placenta. A lot of blood vessels are going to form the fetal part of the placenta. I noticed on the Facebook page somebody posted a picture of a placenta.
That was Margarita.
Yeah, that was from last term. It's an incredible photograph. Y'all ought to look at it.
They're not gonna see it, it's on ours.
Oh, it's on yours.
Can
You
you,
should-
Save the
Yeah, can
photo
you-
and then post it.
I'll ask her. It's for
Well, you don't have to
placenta.
tell who it is. It's just a scientific picture. It's a beautiful picture because it shows this incredible collection of blood vessels growing into a placenta. And the placenta is normally about this big around. It's about that thick, and that's the afterbirth that comes out after the baby comes out. But it's a real thing, and it's amazing that it can have that many blood vessels growing into it, and then they all just kind of separate apart, and the placenta then delivers following the baby, and the woman doesn't bleed to death from it because it's an amazing amount of blood flowing through that thing. Yeah.
Just out of interest, so prior to modern Western medicine, what did they do with placenta and the other accessory organs that are a byproduct of birth? Like, how did they cut the cord? Like, did they even know to do that? Did they wait for it to wither?
Well, I think they probably figured out to cut the cord, because otherwise the whole thing has to dry up, and like the little piece does that is,